Today, we are pleased to present our study entitled A Spatiotemporal Single-Cell Atlas of Porcine Development Reveals Regulatory Dynamics and Cellular Targets of Domestication. The main highlights are as follows. First, we generated a dynamic single-cell/nucleus atlas of pig development, mapping 83 cell types across 5 tissues and 5 stages. Second, we identified regulatory programs driving lineage specification, tissue maturation, and immune diversification. Third, by integrating population genomics, we revealed domestication-associated cellular targets, including MYOT, linked to meat traits. Finally, cross-species comparison with humans showed conserved programs, supporting biomedical research. We profiled 5 tissues, liver, lung, kidney, heart, and muscle, from embryonic day to postnatal day 180, obtaining 252,033 high-quality single-cell transcriptomes. We annotated 83 cell types into 7 major lineages. Cellular composition changed dynamically, muscle, epithelial, and immune lineages progressively expanded, whereas erythroid and stromal lineages declined. PCA and PVCA showed that cell lineage was the dominant source of transcriptomic variation, explaining 46.3%, much higher than tissue origin at 13.6%. This suggests that cellular identity is shaped mainly by lineage rather than tissue context, consistent with strong expression similarity within related lineages. Birth emerged as a systemic transcriptomic inflection point. Neural remodeling peaked around birth, erythroid changes occurred after birth, while muscle cell determination happened before birth. Using P-glycinic regulin analysis, we identified 568 regulins grouped into 8 activity-based modules. These modules represented lineage-specific programs, including immune and erythroid regulation. Regulin activity also shifted over time, such as epithelial cells transitioning from prenatal morphogenesis to postnatal metabolic homeostasis. Together, these results suggest that pig development is driven by lineage-specific TF regulators and dynamic developmental programs. Across 5 tissues, we analyzed 74,572 immune cells and identified 29 immune cell subtypes. Myeloid cells dominated during embryonic development, whereas lymphoid cells expanded markedly after birth. Pseudotime analysis revealed a bifurcated transcriptional trajectory, from an erythroid-like state toward myeloid and lymphoid branches. Consistent across tissues and stages, this pattern suggests conserved architecture for immune diversification. Branch-specific transcription factors included BATF3, IRF8, and SPI1 in the myeloid branch, and LEF1, KLF6, MYC, and IKZF1 in the lymphoid branch, revealing a hierarchical regulatory framework for immune development. We also examined domestication-shaped programs. Integrating Asian and European pig selection signatures with our atlas showed selected genes enriched in neural, muscle, and stromal lineages. Muscle-related signals emerged from E80, coinciding with myogenic differentiation. Functional and QTL enrichment revealed divergent selection involving fatty acid composition, lipid metabolism, fat deposition, body size, and tissue structural integrity. MYOT, encoding a Z-disc-associated protein involved in actin filament stability and sarcomere integrity, was expressed in myogenic cells and higher in European than Asian pigs. Active chromatin at the MYOT locus and Phe links to meat production traits suggest MYOT as a candidate artificial selection target contributing to breed differences. Integration with human prenatal references revealed strong conservation of lineage-specific expression, especially in endothelial, stromal, and immune lineages. Human immune cells showed a bifurcated lymphoid-myeloid trajectory consistent with prenatal pig cells. GWAS disease gene mapping showed conserved enrichment of human disease-associated genes in pig lineages, particularly stromal and immune cells. In summary, this pig atlas reveals mechanisms of lineage differentiation, tissue maturation, immune development, and domestication-associated trait divergence, providing a resource for developmental biology, precision breeding, and disease modeling.